previous videos, I have explained that it is leptin resistance, not insulin resistance, which causes obesity. I have also described how it is the important gut hormones, GLP-1 and glucagon, which are responsible for maintaining leptin sensitivity. And therefore, it is a deficiency of these important gut hormones which is responsible for the leptin resistance, which induces obesity and poor health. And this is why the blockbuster weight loss medications Ozempic and Mounjaro are so powerfully effective. They compensate for the root cause failure of our gut to produce enough GLP-1 hormone by giving a medication which mimics GLP-1. However, as well as or instead of taking the medication, we can endeavour to get our own gut making GLP-1 properly again. And to do this, we need to understand what went wrong in our gut, what made us stop producing enough GLP-1 in the first place, what is the root cause failure. In this video, I will describe that it is toxic bile in the gut, which is blocking the proper secretion of GLP-1 by the gut. This toxic bile is created when there are high levels of bad bacteria and high levels of bile in the gut. The bad bacteria damages the bile, creating this toxic bile. It is very important that we understand the true root cause of obesity and poor health. If we understand what is truly causing our obesity and health problems, that will guide us towards more targeted strategies, which will enable us to optimize our results, instead of what most of us have been doing for a long time, which is blindly stabbing around in the dark, with no accurate roadmap to guide us. Let's get to the science and the evidence. How do the microbiome and the bile conspire to create this toxic bile which causes our obesity and our poor health? It's time to be introduced to the next two very important players. They are the twin brothers, the bile receptors FXR and TGR5. These bile receptors are found in the walls of the gut. Bile receptors in the gut are receptors which are activated by bile in the gut. The TGR5 bile receptor is the good twin. This receptor is the hero of the story. The FXR bile receptor is the villain of the story. The TGR5 bile receptor is the hero because its activation increases the production of GLP-1 hormone by the gut, thereby increasing leptin sensitivity. The FXR bile receptor, on the other hand, is the villain, because the FXR bile receptor decreases the production of GLP-1 in the gut, thereby increasing leptin resistance. Let's look at the evidence that these bile receptors play a very important role in causing obesity and also in reversing obesity. This paper, published in 2020, states that the receptor FXR inhibits GLP-1 secretion in response to microbiota-derived short-chain fatty acids. And researchers in another study reported that intestinal FXR activation impaired energy expenditure and further aggravated obesity and glucose intolerance, while intestinal FXR inhibition blocked FXR, and this exerted metabolic benefits. So, FXR activation reduces energy expenditure and increases obesity, while blocking FXR receptor exerts metabolic benefits. And scientists in another study found that mice bred to lack FXR receptors were resistant to diet-induced obesity, indicating that activation of FXR receptors is an essential part of the mechanism of diet-induced obesity. In addition, another study found that FXR signaling in the small intestine of humans positively correlates with body mass, i.e., people who have more FXR signaling in their small intestines have a higher BMI. In contrast to the negative effects reported for FXR activation, TGR5 bile receptor activation increases GLP production and induces positive effects for obesity and poor health. For example, in this study published in the journal Cell Metabolism, researchers concluded that the TGR5 signaling pathway is critical in regulating intestinal GLP-1 secretion, i.e., TGR5 activation by good bile is the critical regulator of GLP-1 secretion, and therefore it is also the critical regulator of leptin resistance, obesity, and associated poor health. And supporting this, another study published in Cell Metabolism in 2021 also found that the TGR5 bile receptor exerts anti-obesity effects. They reported that increased TGR5 activation by TGR5 activating bile acids causes a reduction in appetite and increased energy expenditure, which are both symptoms of improved leptin sensitivity, and the researchers reported that this decreased the body weight and fat mass of the mice. In contrast, when TGR5 was genetically downregulated, diet-induced obesity was increased. The pivotal role of the TGR5 bile receptor in reversing leptin resistance and obesity is also revealed by new understanding about the mechanisms responsible for the profound weight loss-inducing effects of bariatric surgery. Researchers have discovered that the gastric bypass reverses obesity by improving bile acid signaling. The gastric bypass reduces levels of the bad bile acids, which activate FXR, whilst increasing levels of the good bile acids, which activate the good bile receptor, TGR5. This study, published in 2022, also mentions that it is changes to the gut bacteria that are responsible for the changes in bile signaling. The gut microbiome plays an important role in the development of obesity and poor health because it is the gut microbiome that controls what type of bile we have in our intestine and brain. It is our gut microbiome that controls if we have good bile or bad bile. The next question is how? How does the gut microbiome control the type of bile that we have in our intestines? It is because the problem gut bacteria damage the bile. So, when we have an overgrowth of problem bacteria in the gut, those problem bacteria damage the bile, resulting in high levels of this damaged bile, this bile that's been damaged by the problem bacteria. And this type of damaged bile is the bad bile that activates the FXR receptor and reduces GLP-1 secretion. On the other hand, when we have a well-controlled gut microbiome with low levels of the problem bacteria, there is no or minimal damage to the bile. This undamaged, healthy type of bile is the good bile. It is the bile which activates the TGR5 bile receptor and increases GLP-1 secretion in the gut. Researchers investigating interactions between gut bacteria and bile concluded from their experiments that the gut microbiota promoted diet-induced obesity, adipose tissue inflammation, and liver inflammation, and that this occurred in a strict FXR-dependent manner, i.e., the activation of FXR is an essential part of the mechanism by which the gut microbiota causes obesity and poor health. They state, our results support the concept that the gut microbiota promotes diet-induced obesity and associated phenotypes through effects on the bile acid profile and altered FXR signaling. And in another study published in Microbiome journal in 2023, researchers similarly concluded that intestinal FXR activation is required for the gut bacteria to induce high-fat diet-induced obesity, insulin resistance, and other poor health symptoms, i.e., FXR activation is an essential part of the mechanism by which gut bacteria induce obesity and poor health. And more studies have clarified the mechanism of how the gut microbiome induces FXR activation. This study led researchers to conclude that microbial modification of bile in favour of bile acids which activate FXR is the mechanism by which the gut microbiota promotes diet-induced obesity and associated phenotypes. So they found that certain gut bacteria modify, change, or damage the bile, and that this changed, damaged bile increases the activation of the bad bile receptor, FXR. Other studies have clarified how the problem bacteria modify the bile in this negative way to create this bad bile that activates FXR. For example, researchers in this study concluded that through defined enzymatic activities, the gut microbiota can significantly modify the signaling properties of bile acids and therefore have an impact upon host health, i.e., they are saying the gut microbiota modifies or damages bile through enzymatic activity, i.e., the action of the bacterial enzymes on the bile, and this creates this modified, damaged type of bile, which increases FXR activation. And more studies have even shed light on what these bacterial enzymatic activities are. How does the enzyme activity of the problem bacteria turn the bile into this bad FXR activating type of bile? What happens is that certain problem bacteria produce an enzyme which modifies the bile. This enzyme is called selective bile salt hydrolase. This bile salt hydrolase breaks down the bile. Now, this selective bile salt hydrolase enzyme only breaks down the good bile. It only breaks down the good TGR5 activating bile, but it leaves intact the bad FXR activating bile. The result of this bile salt hydrolase enzyme selectively only breaking down the good bile acids results in a bile acid pool which is rich in FXR activating bile, but depleted of TGR5 activating bile. It is a bile acid pool which is rich in the bad type of bile and low in the good type of bile. And the result of this is greatly reduced GLP-1 secretion in the gut, which results in leptin resistance, obesity, and poor associated health. Let's look at the science which supports this mechanism. In this research article, researchers reported that mice colonized with bacteria unable to produce this problematic selective bile salt hydrolase enzyme gained less weight on a high-fat diet and had lower levels of fat in their blood and liver. They also shifted to burning fats instead of carbohydrates for energy. This is another symptom of leptin sensitivity. So mice which were unable to produce this enzyme, which breaks down the good bile, had less body fat and were more healthy. This strongly implicates the important role of this problematic selective bile salt hydrolase enzyme in the induction of obesity and associated poor health. So, to summarize, leptin resistance and obesity is caused when there is an overgrowth of problem bacteria in the gut, which damages the bile in the intestines, leading to increased activation of the bad bile receptors and decreased activation of the good bile receptors, which reduces GLP-1 secretion, causing leptin resistance, causes obesity and associated poor health. The evidence I've presented here is just a small amount of the scientific evidence, which continues to mount daily. In my next two videos, I will show you the evidence that all the popular diet strategies like intermittent fasting, low-carb diets, low-fat diets, high-protein diets, and whole food plant-based diets all work to reverse obesity and poor health because they reduce the production of bacterially damaged toxic bile. i.e., this is the real mechanism responsible for why these diets all help us to improve our health and obesity. They don't work because they're reducing insulin spikes. They don't work because they're reducing glucose spikes. They don't work because they're reducing intramyocellular lipid storage. They all work because they reduce the production of bacterially damaged toxic bile in the intestine, which is the real root cause of obesity and poor health. What do you think of this new model of obesity? Does it make more sense to you than the old models? I would love to hear your thoughts in the comments. Thank you for listening and hopefully see you in the next one.